Today, we would like to talk about multivariate pattern analysis in neuroscience. You know that these patterns of activations of neural dynamics is what really matters in our field. So, is what we really needs to look for. Imagine that we have an fMRI, it could be also an EEG or MEG, and we have information from the brain. This information, for example, in the case of fMRI, comes in little boxes, little pieces of information that are describing our brain activity. So what we are trying to look for, we're trying to look for which one is the specific activation patterns that are reflecting each one of the cognitive processes we are trying to understand. So first of all, imagine that we take one little point of this huge brain, and this brain and this voxel, the first one, is indicating how much activity it has for one trial A, let's say the condition A, or let's say the condition B. Once we have all the conditions and we have the information for this voxel, we can, for example, do a histogram. This histogram could understand, hey, how much it distributes over the voxel the activity of this specific voxel for the condition A or for the condition B. So this information is super useful, and this is how univariate model works. Let me say that now we take the mean, the average of each one of the groups, and once we have the average, we can describe the probability distribution, the density distribution, and with these density distributions, you already know how to do statistics. So we can compute which one would be the p-value of this first box. But here today, we are not going to approach this way. Otherwise, in other way, we are going to talk about multivariate pattern analysis. So this involves more than one voxel. So imagine that we take now a second voxel from this brain. The second voxel also has A activity, B activity, condition A and B, and this activity distributes in this 2D, two-dimensional space this way. So what we can see here is still the same density probabilities, the same histograms that we saw at the beginning, but also now we have another two histograms for the second variable, the voxel 2. What's the main difference of what we want to do here? Is that here, imagine that we are going to establish a line, for example, this yellow line, that is going to indicate, okay, whether you can split your points in A condition and B condition. Actually, what we are doing here is exactly the same that we were doing in the previous uh, analysis. But instead of an univariate pattern analysis, where you can just identify the distribution of the two data, the two conditions in the same voxel, here we have two voxels and each voxel is gonna provide some information. So here, if you can see, we have also the same two distributions for A in blue and for B in red, but the multivariate is forcing the mean, the average of its distribution to be slightly different, slightly more open, and as a consequence, in terms of statistics, we could say that we have the prima, the difference or co-NSD more open than in the case of the univariate. So this is the main point of what we are trying to do in a multivariate approach. We try to move both distributions, to separate them, to better and more clearly understand what's going on in each one. This is the main important thing. and we are achieving this by using, for example, a linear discriminant analysis, a supported vector machine, neural networks, or any kind of decoder that handles this more than one input, and its output, it indicates which one is the label. Let's call label A or label B. As a consequence, we have a better discrimination of the activity. But now imagine that we have even a third volume. We have a third dimension, we have a third voxel, and this new dimension is gonna even provide more information. In this case, instead of having line, that this line is dividing our sample, our 2D sample, now in the case of adding 3 dimensions, we have to create a plane. So this plane is indicated. Everything that goes above the plane is called A. Everything beneath the plane, deeper plane, is going to be B. But actually, it's the same, just adding this new information. If we have more voxels or more channels in the EEG, or more channels in the MEG, or more time points in the time domain, as we will see, is like you are increasing the information you have. So the idea is to leverage this n-dimensional space and try to understand better how this dimensionality works in the brain. Why? Because what we want to do is to measure, to understand, to find these neural patterns. And these neural patterns appears in the brain. Imagine that we have voxel 1. For simplicity, here, instead of having a continuous line of values, for simplicity, let's say we have no activated, 0 or 1. Okay, so how many patterns do we have with one voxel? With one voxel, if we do an univariate approach, we could have only 2 patterns, 0 or 1, no activity or activity. Of course, this is reductionist. We have more than 2 patterns. Actually, as far as the voxel is is a continuous variable, we have more information. But for simplicity, let's stick to it. But what happens if if we take into consideration a second voxel? We take another voxel. This second voxel is creating more patterns. So we could have, for example, 0, 0, no activity, no activity, 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 no activity, 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 no activity. So now the possible patterns, taking into account these 2 voxels, it increases until 4 possible patterns. And what happens if now we talk about a third volume? Only with 3 voxels, the number of possible patterns, it increases and rises until 8. As we could have no activity 0, 0, 0, 1, 1, 1, 1, 0, 1, 1, 0, 0, multiple patterns. And for example, if we go to more and more, we will raise more information. Actually, here what we are talking about is that the possible patterns are going to exponentially grow as a matter as the power of 2 elevated to voxels, the number of voxels. The more voxels, the more channels in EEG, the more time points in MEG, the more information multivariate, in a multivariate way we are increasing to the analysis, the more information we're going to have. And this is why, talking about the brain, we should work with neural patterns, because these neural patterns in fMRI, in EEG and MEG, in arrays, intracorticals, in any kind of information, is giving and providing this rich information, as you have seen, splitting more and clarifying the differences between 2 conditions, and also increasing the information.